Hello, welcome to video 2 of the gene regulation in prokaryotes. Now, I should say that a lot of this stuff here is not just for prokaryotes. This is we're getting you into it. We're getting you introduced to what gene regulation is with its, as this slide shows, modulators and repressors, what an operon is, things like that. We talked a little bit about the tryptophan one, but here is the lactose or the lac operon, which it's an example of an inducible operon. That is one that can be turned on by a particular modulator that inhibits the repressor. So it can be induced to being turned on when lactose is present. When lactose is present, we need to have the ability to break it down. And that's what these genes do, these genes right here. And when lactose is present, we can inhibit the repressor, thus activating the operon. So this is an inducible operon, and there's a slide here which is illustration of it. And these slides keep talking a little bit about the regulatory gene that builds the repressor, and then there's animations of that. And if you have an inactive a repressor, then you can go ahead and build the proteins that'll help us break down the lactose. We moved on to the tryptophan or the tripoperon here, which is a little bit different from the lac operon. The lac operon, if you remember, was a inducible operon. This is a repressible one, which means it's on all the time unless too much tryptophan is present. So essentially you're repressing this. If you're not turning it on, it's always on. The lac operon is not on unless you turn it on by having lactose present. So repressible operon versus inducible one, that's a big difference. And we have the same sort of animations and things that can kind of teach you about how the operon works and the different parts of it here. But both of them are examples of negative gene regulation, as the repressor protein inhibits transcription of the operon. So one way or another, these are negative loops. And negative gene regulation is very common in prokaryotes, but there are some examples of positive ones. And this is an example of a positive one here, and it's going to be talking a little bit more about how we can get this protein here that actually helps RNA polymerase attach. And you can see how it works here. And I want to mention that almost all of this stuff is allosteric, which means the shape of it, the actual shape. Remember how we always said shape always relates to function, and that's definitely the case with these. Now, when you get done with this presentation, and I didn't explain everything on it, just touched on some things. Once again, understand we are getting into gene regulation. How and when are our genes turned on? When are they turned off? How do our genes know to make skin on the outside? Or how do they know how to make bones? Or how do they know how to put our head towards the interior and our tails towards the posterior? All kinds of that stuff. After this, getting through with this presentation, you need to watch these videos. That'll help give you some more extra explanation. And then after you're done with that, you need to make sure to go back. And the first thing you have is the Gene 1 regulation assignment. It's due March 26th at 8:40 a.m. You're going to have to do it essentially like a pre-test of sorts. So you're probably not going to know everything, but do your best, because we're going to eventually use that, and it'll help you with the actual test later on. Use your test-taking strategies as best you can. Good luck. We're praying for you. Stay healthy, and miss you.